So I'm going to start sort of sketching in the corner here, but if you're working, keep going. So hopefully we have something like the cortex of the kidney up here. Got our medulla down here. Hopefully you'll be seeing these in lab. These are actually visible differences in the kidney. We got our loop of Henle coming in, right? We had our proximal tubule and the cortex. Loop of Henle diving down into the medulla and then coming back up. So here's our tubule. So what I'm drawing now is the descending part of the loop of Henle. Then we have a thin part of the ascending limb, and then a thicker part. Eventually we're going to go into our distal convoluted tubule, and then into a collecting duct. We'll be talking about the distal tubule and that collecting duct, which actually receives fluid from many nephrons today. So the loop of Henle, it's important for really is establishing and then maintaining our osmotic gradient, by which we really mean that we have a difference in kind of how salty the medulla is, starting at about 300 milliosmoles at the top. That's also our osmolarity of the cortex and of the fluid before it goes into the loop of Henle. Then that medulla, to allow for continuous reabsorption, increases to 1400-ish milliosmoles. We descend deeper and deeper into the medulla. So really the reason we have this gradient is that we have a difference in what the descending limb versus the ascending limb is able to do. So this is actually, if you sort of step back and think about it, due to differences in the cellular membranes, so the plasma membranes of the cells that form the descending versus the ascending tubule. So in the descending tubule, they're permeable to water. So water can get out, H2O. And in the ascending limb, we are pumping solutes, right? So sodium, potassium, maybe some chloride. So this is important to setting up the gradient because pumping those solutes is part of what's setting up the high osmolarity of the medulla. Okay. So a better drawing looks something like this, right? So water coming out of the descending limb, so it's permeable to water, but it's not transporting any of our solutes, sodium, potassium, chloride ions. Our thick ascending limb is transporting all of those things, right? But it is not permeable to water. It's impermeable to water. The other solute that we want to remember that is actually really important to this as well, contributing to about 40% of the osmolarity here, is urea. So urea is building up in the medulla as well, and that contributes as well to this medullary osmotic gradient. Just a quick reminder, because that was at the end of yesterday. I'm not going into super deep detail on urea, but the reason that urea is able to cross into these areas is that we have urea transporters in specific places. So we have urea coming out of the collecting duct. So that's getting into the medulla. What we'll notice is that the descending limb of the vasa recta, which is the capillary system surrounding the loop of Henle, is also going to have urea transporters. So it's actually absorbing some urea as well. So this eventually is going to lead to the fact that as the vasa recta, as the blood flow leaves the kidney, as it leaves the medulla, it's going to be a little more concentrated. It's going to have slightly higher osmolarity than it did before when it goes in. And that's going to be on a slide later, but I'm just showing you now while we have a picture of the vasa recta up. This is a picture from last time. So just a reminder of how blood flow is going through here, because that's what we're going to be talking about at the beginning of today. So we have our afferent arteriole coming in. It gives way to the glomerular capillaries, right? So the glomerular capillaries have basically very porous walls, allowing everything to kind of get bulk filtered into the nephron. Our efferent arteriole leaves the glomerulus. It's skinny, it's narrow, so that we can kind of build up blood there in the glomerulus, because we want to force lots and lots of blood to kind of get filtered into the nephron. Not blood getting filtered, but the contents of the plasma, right? We're filtering a lot of that into our nephron. As the efferent arteriole leaves that renal corpuscle, Bowman's capsule, the glomerulus, those are the parts of the renal corpuscle. It gives off our peritubular capillaries. So these are surrounding both the proximal and distal tubules. Mainly talked about them as regards the proximal tubule thus far, but this is our first site of reabsorption of solutes and water. Right, so we have solutes and water going into those peritubular capillaries. Reabsorption is when we go from our proximal tubules or our distal tubules, really wherever, into the blood, back into the blood, allowing it to circulate. So here we see those peritubular capillaries joining back up, right, because they're continuing with our blood flow. And then we enter the vasa recta, called vasa recta. Vasa, right, comes from, sounds kind of like veins, right? Vasa, I don't know. Vasculature, right? One of these words telling you it's blood flow. Recta, telling you it's kind of up and down, kind of a straight vessel. So the vasa recta surround the loop of Henle. What I want you to notice before we go through a couple slides on the vasa recta is that they're going the opposite direction of the flow through the loop of Henle. So if we trace the flow through the loop of Henle, right, we have descending limb over here on the left, looping down, coming up is the ascending limb, right? But if we're tracking the blood flow in the vasa recta, you can see that it's coming down against the flow of that fluid that I just traced in green, and then returning along the descending limb of the loop of Henle. So when we refer to the descending limb of the vasa recta, we're referring to this part. So the descending limb of the vasa recta is next to the ascending limb of the loop of Henle. Right. And the ascending limb of the vasa recta is next to the descending limb of the loop of Henle. Yellow's not very visible, but just want you to note that opposite direction. So that's what's going on here before we talk a little bit about what's happening in those physiologically. Okay. So the role of the vasa recta in the medullary osmotic gradient is basically to help this gradient from dissipating. We don't want to even out everything in the medulla. So we've established this osmotic gradient. We've put in some effort to create it. So the shape of the vasa recta, we need blood flow there, right, in order to allow some reabsorption, things like that. But we don't want to water down these deeper parts of the medulla, and we don't want to increase the salinity up here at the top of the medulla in particular either. So the anatomical arrangement of the vasa recta allows for that. So Basically, the fact that it has that kind of hairpin turn at the bottom means that the blood through the vasa recta is moving pretty slowly. So that's one of the things that's helping us not sort of diffuse this gradient and reach equilibrium everywhere. And also the fact that it's flowing in the opposite direction from the flow through the loop of Henle also has an effect there. So we're going to keep it a little simple with our understanding of the vasa recta. Basically, what I want you to know here is that the descending limb of the vasa recta, so descending limb, and here in this picture, it's showing you the vasa recta. I know it looks like a nephron, looks like that loop of Henle, but this is specifically just showing you the blood flow, even though they've colored it yellow like we did before. So the descending limb of the vasa recta, which is next to our ascending limb of the loop of Henle, is permeable to water. So we have water leaving that descending limb of the vasa recta. We have solutes entering it, all right? So it actually has solutes and water moving through it, right? So that's what we see happening here. Water leaving, solutes entering. All right, so we might think that that water would water down our osmotic gradient. But fortunately, unlike in the loop of Henle, where we have one limb permeable to the water and the other limb impermeable to the water, in the vasa recta, both of them are permeable to water. So water is moving out in the descending limb, but then it just moves right in to the ascending limb, essentially. These things are kind of close together. So while it does sort of start to water things down, it then just gets reabsorbed back into the vasa recta on the other side, the vasa recta that's coming up, because of the slightly increased osmolarity, which is partially due to that fact that we have urea moving into the vasa recta down here, as we were looking on that previous picture. Right, so the osmolarity is higher because urea can't get out of the ascending limb. So it's not saying that none of the vasa recta have urea transporters. It's saying that the urea can't get out of the ascending limb. So the fact that that blood flow has a slightly higher osmolarity here means that we just reabsorb that water again. So the net kind of water movement across the descending and ascending limb of the vasa recta is pretty much zero. So that's why we're not kind of erasing this gradient. Does that make sense to everybody? Any questions about that? Okay. So to this point, we've talked about water reabsorption. Ooh, didn't mean to pause that, sorry y'all. So we talked about water reabsorption in the proximal tubule, all right? So this is where we had our mass reabsorption, our bulk reabsorption. It wasn't regulated, by which we mean we didn't have like hormones coming in and changing how much water is being reabsorbed in the proximal tubule. We're just filtering out, it's following solutes, it's following gradients. Okay, so that's what's happening in the proximal tubule, lots and lots of water being reabsorbed. We've talked about water movement with the loop of Henle, but what I'd like you to realize is that as we talked about water movement in the loop of Henle, what we were really talking about is setting up gradients in the medulla rather than reabsorbing a bunch of water into the blood flow itself. And that medullary osmotic gradient is important for water reabsorption, but the place where we actually have the water flowing into the blood substantially is in the distal tubule and in the collecting duct rather than in the loop itself. So the loop is setting things up for reabsorption, but it's not a significant site of reabsorption itself. So that's why this is the loop of Henle isn't on this list of where water is being reabsorbed. Okay. So the remaining 30% of the water that we're reabsorbing, 20% is going to be reabsorbed in the distal tubule. Okay. And then 10% is going to be reabsorbed in the collecting duct. So those ducts that are collecting, basically at this point, urine from several nephrons. As we talk about the distal tubule and the collecting duct, we're going to be paying attention to this hormone, ADH, or antidiuretic hormone. So we say that the reabsorption in the distal tubule and the collecting duct is regulated, right? It is regulated because it's all basically going to be controlled by ADH. So some of the time we're going to reabsorb a lot of water in the distal tubule and the collecting ducts. Sometimes we're not going to absorb very much at all. And that's dependent on levels of ADH. So that's an important way that they differ from the reabsorption we saw in the proximal tubule, where the water was just following basically the ions that we were reabsorbing. And so we're going to go into some detail on ADH and how that works. So the distal tubule and the collecting duct, do them kind of at the beginning, right? But we remember we have our cortex, right? We have our cortex, we have our medulla below it, right? So like this. When we're following our nephron, we have our proximal tubule, we have our loop of Henle, we're coming back up towards the cortex for the distal tubule, go a little bit into the medulla, and then into a collecting duct. Collecting duct is receiving urine from several different distal tubules. Okay, so what we want to note here is that our distal tubule and our collecting duct, right, they are here diving into the medulla, right? So when we say that the loop of Henle sets up the osmotic gradient in the medulla, this is what it's setting it up for. It's setting it up for when the fluid then passes back into the medulla in the distal tubule and the collecting duct. So more formally, we say it's dependent on the osmotic gradient established by the countercurrent multiplier. That countercurrent multiplier, right, was the loop of Henle, the fact that we have water coming out in the descending limb, but we have a bunch of ions being pumped out in the ascending limb. Okay, so but now we're not focusing on the loop of Henle, we're focusing on the distal tubule and the collecting duct. So we're thinking about water reabsorption, and we're going to realize that how we kind of toggle how much water is reabsorbed, it's going to be dependent on how permeable that epithelium is to water. So some of the time we're going to see a membrane where water can cross, easy peasy, right? And it's going to cross into blood flow that is surrounding this collecting duct and the distal tubule, right? So I haven't drawn on the blood flow, but that's where it would be going, reabsorbed into the bloodstream. But some of the time, we're going to be able to turn off that permeability, right? So we're going to turn off the transporters that are allowing the water to cross, or basically, we're not going to be putting transporters in there in the first place. When we don't have these transporters called aquaporins present in that membrane, not much water can cross the membrane at all. So how much water we reabsorb is dependent on the transporters in the membrane in these areas. So that's what we're going to be toggling back and forth with ADH. And how much water we're reabsorbing, just to kind of really step back and think about what this means kind of in a clinical setting, how much water we're reabsorbing is really going to have an effect on how much urination is happening. So if we reabsorb a bunch of water, right? So if we do allow a lot of water to leave the collecting duct, What do you think the effect on concentrations of waste products and solutes is going to be here? And on the amount of urine. If we have water leaving, we're going to end up with really sort of concentrated urine, right? Like lots of solutes in that urine. And we're going to end up with a low volume of urine because that water got reabsorbed. That water left, it went back to the blood. And the opposite is also true. If we turn off those transporters and we don't allow water to cross, that water is going to stay in the tubule, going to stay in the collecting duct, and so it's going to show up in the urine. So we're going to have a really high volume of urine if we make this membrane impermeable to water. Now, once you kind of internalize that fact, that relationship will seem obvious, but I don't know, the first time I learned it, realizing that sort of balance between urine concentrations and volume, these transporters took a little time to wrap my mind around it. But that's where we're headed. That's kind of the direct effect that you would observe of this membrane permeability. Okay. So that water permeability is dependent on channel proteins for water specifically. So often we talk about membranes as being permeable to water when you first learn about cell biology, but actually what makes membranes really permeable to water often is the presence of aquaporins. So aquaporins are fairly common, but when we're talking about the kidneys, we need to be really precise about where they are and where they are not. Okay. So we have two types we're thinking about as we think about the distal tubule and the collecting ducts. So we're thinking about aquaporin 3 and we're thinking about aquaporin 2. So aquaporin 3 is always going to be present, and it's specifically present in the basolateral membrane. So if we have our cell, right, drawing an apical membrane, I'm just going to draw some microvilli to indicate that to you. Not every cell has microvilli, but it makes it identifiable. Basolateral membrane is the one that's right next to blood flow. Okay. So here's our blood flow. Okay. So aquaporin 3 is always going to be present here in the basolateral membrane. It's not always going to be present. We're not always going to have aquaporin 2 on our apical membrane. Right. So here we have a question mark. Maybe it's there, maybe it's not. It's going to be dependent on ADH. Questions about this so far? We are going to look at the role of ADH in controlling these aquaporins in a second, but are we good so far? Okay. Okay. So when we're in this late distal tubule, it's specifying late distal tubule because that's the part that's in the medulla, right? So that is the part where we would expect to potentially see water reabsorption, or in the collecting duct, which is descending all the way through the medulla. When that membrane is impermeable to water because it lacks aquaporin 2, that means that water is not going to be able to leave the tubules because it has to cross both membranes, right? For water to get reabsorbed, it needs to get across the apical membrane using aquaporin 2, and then it needs to get into the blood using aquaporin 3. So if we don't have aquaporin 2, water can't leave the tubule in the first place. It can't get in the cell. So it doesn't matter that aquaporin 3 is present because that's only on one side of the cell. So if we don't have that aquaporin 2, means we're going to have no water reabsorption because no water is going to go into those cells, which means that more water is staying in the tubules. And the tubules are producing that urine. So if water stays in the tubule and doesn't get reabsorbed, becomes urine, we end up with a high volume of urine. The way we get aquaporin 2 into the membrane and make those membranes permeable to water is through this hormone ADH. What ADH does is it creates an impetus for the cell to insert these water channels, the aquaporin 2, into the apical membrane. And when they are present, water is going to be able to be reabsorbed by osmosis because it's going to be able to cross both membranes, both the apical and the basolateral. The maximum urine concentration is going to be about 1400 milliosmoles. So like there is a cap on how much water you can reabsorb, because in order to actually have a urine flow and actually get rid of waste products, you do need something for it to flow in. There's actually an obligatory water loss of about 440 milliliters. And the maximum urine concentration in that 440 milliliters would be about 1400 milliosmoles. So that same number that we've been seeing in the medullary osmotic gradient. And the maximum amount, really, that water can be reabsorbed depends on the length of the loop of Henle. Basically, all that means is that if you have a longer loop of Henle, you are setting up more and more of an osmotic gradient. Like as it dives deeper, you set up more and more osmolarity. So if you have a long loop of Henle, at the bottom of that loop of Henle, you're going to have a really high osmotic gradient. And this varies in some other species other than humans, right? Like camels have really long loops of Henle, allowing for a lot of water reabsorption. But loops of Henle are really variable. Nephrons are variable. We've been looking at a very kind of stereotypical nephron, but in your own body, you'd have multiple different types of nephron, different lengths of loops of Henle. All right, so here we just see a picture of what we've been talking about. All right, so here we have a tubular fluid coming along. Our late distilled tubule here is impermeable to H2O. It's entering the collecting duct. This late distilled tubule is actually entering the collecting duct up here in the cortex. And we have multiple sort of places the late distilled tubule can join in. But importantly, that collecting duct is diving into the medulla. But here on the left, we see a collecting duct that is impermeable to H2O, right? So even though we have a gradient, right? So we have a gradient, right? Identifying this gradient, right? We have, say, 100 milliosmoles here in that fluid, that urine. We have 600 milliosmoles here in the medulla. So water would want to flow in this direction, but it can't because we don't have the right transporters. We don't have that aquaporin 2. We have a difference here in our image on the right. So on the right, we're seeing a collecting duct and even a late distal tubule that is permeable to H2O. So permeable, even in this distal tubule, right? Even though we're in the cortex, we still have a gradient. 100, this whole cortex is 300, so we have a gradient. 100 is less than 300, so that 300 is pulling on the water, so water leaves our distal tubule. As we go into the collecting duct, we're tracing a collecting duct that is also permeable to water, so it does have that aquaporin 2. So instead of keeping that osmolarity of 100 all the way through, we can see that as it descends, that fluid is reaching equilibrium with the medullary osmotic gradient. So it would have kind of started at 100. We have water leave, so we can see that that tubular fluid that was at 100 milliosmoles reached equilibrium with the cortex value of 300. As it goes down, it's increasing because it's reaching equilibrium with the medulla because we're allowing water flow. So the result here, when we allow water to get reabsorbed, is low volume, but highly osmolar, so highly concentrated urine. Versus if we don't have those aquaporins, lots of water stayed in our urine, so it's high volume. And it's not very concentrated, it has low osmolarity, because it didn't go through this process of reaching equilibrium with the medulla, because there was no way for the water to cross. A questions about this kind of high level difference, this impermeability versus permeability? Okay. So I mentioned there's an obligatory water loss, 440 milliliters. The osmolarity of that urine would be about 1400 milliosmoles, because that's how osmolar the medulla gets at the depth of the loop of Henle. So this is true for humans because that's what we're talking about in A&P. Some solute is excreted in that urine. Okay, now we're going to think about the actual control of that permeability. So we're thinking about the control of water balance and the osmolarity by antidiuretic hormone, right? Control of water balance. Remember, water balance means how much water is going in to you, plus what you're producing, how much water is going out of you, plus what you're using up. So really, we're controlling excretion here by controlling the volume and osmolarity of that urine. So we're controlling the out here. That's why this relates to water balance. We're doing that with antidiuretic hormone, ADH. Okay. So ADH is regulating the permeability, both the later part of the distal tubule and the collecting duct. So what it's doing is it's varying how many aquaporin II transporters we have in these areas. Depending on how much ADH there is, we can, it's not an on or off switch, I guess is what I want to say to you. So we can vary the urine osmolarity in a range from 100 to 1400 milliosmoles. It's not one or the other. So we can kind of increase the number of aquaporin IIs, or we can decrease the number. So in real people, it's not like you would be completely impermeable or super permeable. You have varying levels. So these are really ends of the range, 100 and 1400. So you may have heard the word diuretic before, right? So you may have heard that like coffee and tea are diuretics, right? So a diuretic, something that makes you pee, basically. So when we say a diuretic, we think of people peeing a lot. So that would be high volumes of urine, right? That's why you pee a lot, is your bladder is filling quick. So antidiuretic means that the presence of ADH is going to have the opposite effect. So you're not going to pee that much. You're going to have low volumes of urine. The reason for that is you're reabsorbing that water. That water is going back into your blood. Ooh, my accent came out there. Water is going back into the blood, which means it's not in your urine anymore, which means you don't have that much urine, because the main part of your urine, right, is the water, right? That's what's making up the volume. Okay. So here's a diagram of what ADH is doing, and we're going to kind of step through this. Okay. So ADH is a hormone. It's actually coming from your pituitary. So remember way back when we talked about the pituitary gland, the endocrine system, ADH is one of the hormones coming from your posterior pituitary. And we'll talk about how ADH production gets stimulated in a second. But ADH is going to end up in your bloodstream, right? Which means it's going to go through the peritubular capillaries next to your distal collecting duct, which means when we have this hormone going through the capillary, it can leave the capillary and go affect our cells. All right, so ADH carries through in the plasma coming from the posterior pituitary, and it comes up, bother our cell, When I say bother, what I really mean is we're binding to a receptor for ADH. So a receptor for ADH means we have a protein embedded in this basolateral membrane. The basolateral membrane is the one on the blood side. So we're binding into that ADH receptor. It happens to be a G protein coupled receptor. So this G protein attached to our receptor kind of communicates that signal to some other proteins embedded in the membrane. So specifically, we have this enzyme, adenylate cyclase, which causes us to create our second messenger, cyclic AMP. Okay. So step one was we bound to the ADH receptor. Step two is that the receptor essentially creates cyclic AMP. And there's a little intermediary step, but that's where we're going. That's our second messenger. Then we have kind of a flow of reactions. Specifically, cyclic AMP triggers protein kinase A. Right? What protein kinase A does is it communicates a signal to these vesicles, or these kind of little baggies in the cell, that are holding a bunch of aquaporin II transporters kind of ready to go. So those proteins have already been made, they're being stored in the cell, and the cell is just waiting for this signal from ADH. And so it's waiting for this signal to get into the cell, pass through the series of signaling molecules inside the cell, get to that vesicle, and tell it to insert into the apical membrane. So really the parts I care about this for you to know, it's important to kind of understand how this works, but what we want to know is ADH getting into the cell is first step, all right? And then we want to know that we have this vesicle that is ultimately triggered to release those proteins that it's carrying and insert them onto the apical membrane. So the important parts for you to remember are the beginning and ending of this process. Okay. So ADH triggering this pathway, vesicle then releases aquaporin II and it gets embedded on the membrane. Now when aquaporin II is present, water is able to flow through the cell, right? So water goes through these aquaporin IIs that we just inserted, right? So now it's in the cell. And then water, once it's in the cell, passively flows down its gradient into the paratubular fluid and ultimately into the plasma on the other side. So the second part that it was flowing through was that aquaporin III, so that's the one that's always present, right? So water can always basically get from the cell into the blood. It just can't always get from the fluid into the cell. Right? So this is the part that we're toggling on and off with our ADH. Questions on this diagram before we move forward? Okay. Okay, so the way that ADH gets into the plasma is through the endocrine system. So specifically, it's coming from the posterior pituitary, which we remember is up in our brain. Now, when we talked about the posterior pituitary, we talked about the fact, right, there's an anterior pituitary, the front half, posterior pituitary, the back half of that gland. Now, the posterior pituitary is the one where we basically have neurons originating in the hypothalamus, and directly the other end of the neuron, the terminals, are in the posterior pituitary. So when we talked about like neurohormones, ADH was one of them. So this is kind of a throwback, but I know that endocrine unit was a lot, so it's worth a reminder. So the way your body knows to secrete ADH is that we actually have receptors up there that can sense the osmolarity. So basically they're sensing the osmolarity in the blood. Usually we have a blood-brain barrier. So usually most parts of your brain would not be able to do this. It's just this specific part, the OVLT, organum vasculosum of laminae terminalis, OVLT, has osmoreceptors. Osmo means they're sensing osmolarity. Receptor means they're sensing something. So what they're sensing, it's like how much solute is in your blood, basically how salty is your blood, right? So when they sense that you don't have enough water, right, to make the plasma an appropriate concentration, it's going to tell you to reabsorb more water so that you preserve that in your plasma. Right? So that's what the osmoreceptors are doing. ADH is also going to be affected by baroreceptors detecting blood volume and pressure. So remember that we have those receptors for blood volume in the veins. We have our MAP receptors in the artery. When we have low baroreceptor activity, so low baroreceptor activity basically tell us like we don't have enough blood, right? Our blood volume is too low, we're getting dehydrated. That's going to up our ADH secretion. It's going to tell us get more water in the blood, do more reabsorption, right? So ADH tells you to reabsorb water into that plasma, increasing plasma volume and decreasing its osmolarity. So here we have basically our two triggers for ADH release. So the bottom of this image is going to be similar in the next two diagrams, right? So the bottom part is that if we increase ADH secretion, our kidneys are going to absorb more water because of those aquaporin 2s that we're putting into our apical membranes. If we reabsorb water into the bloodstream, it's not going to be in our urine, so we decrease water excretion, and overall we're conserving our body water, right? So we're going to be in positive balance basically for water. We're going to be increasing that blood volume. This could be a negative feedback loop on whatever our trigger was. So in this case, for this image, what we're looking at is how our osmolarity, or basically the concentration of things in the blood, is affecting ADH secretion. So here we have our osmolarity increasing. So say you have really salty blood, got lots of sodium in it, high osmolarity. Osmoreceptors detect that. Hey, our blood's too concentrated. In the hypothalamus, increases the neurosecretion, right? Those cells are going to secrete our ADH, leading to this whole flow. Very similar thing is happening with our baroreceptors, right? So this whole bottom half of the diagram is the same thing, right? More ADH, more water reabsorbed, which means it's not in the urine. Water's not getting excreted, so we're conserving water in the body, water in the plasma, which means we're also conserving blood volume. The thing we're trying to affect is that we've sensed low blood volume from the veins. We've sensed low blood pressure from the arteries, okay? Means we don't have enough blood. Baroreceptors are detecting this. Same deal, right? The only difference here is that we don't need to affect those osmoreceptors. We're just telling the hypothalamus this directly, increasing the activity of those cells in the hypothalamus. The final thing we want to think about here with our water excretion is that glomerular filtration rate also does have an effect, right? We talked a bit at the beginning about how glomerular filtration rate is regulated. We talked about those regulatory mechanisms, right, for keeping glomerular filtration rate in this nice zone to keep us with these to correspond to our usual mean arterial pressure range, right? But if blood pressure is dropping outside of this normal range, we do allow the glomerular filtration rate to vary. So it would not be regulating us back to our 125, our usual level of glomerular filtration. So if our blood pressure drops really low, right, so we drop below 80, this means we don't have that much blood going through the glomerulus. We don't have that much blood, means we don't have that much hydrostatic pressure pushing out on those capillary walls, which means we don't have that much water getting filtered into the nephron in the first place, because that's one of our Starling's forces for filtration. So we decrease glomerular filtration rate, decreasing our water filtered. If that water never goes into the nephron, it's not going to be excreted, right? So if we have a super low blood pressure, we're going to decrease our water excretion, which means we're going to retain water, right? Which is actually kind of good for us, right? We don't want to have that low blood pressure. We want to conserve our body water. If we go up above 180, and we should not go above 180, but if we do, this increases that hydrostatic pressure in the glomerular capillaries, which means more water is going to get filtered out, right? Because we have lots of force for filtration. We have water pressing out, pushing into Bowman's space. Because we have lots and lots of water in the nephron, we're going to excrete more water. So we're going to end up with more water in our urine just because we're pushing lots of water through fast. This would tend to kind of bring this back down potentially, but this is actually only really occurring in pathological circumstances because we shouldn't be up that high. Okay, so we are going to leave it here for today, and next time we'll pick up with our electrolyte balance 2 packet. All right, see you all Friday. Have a great rest of the week.